Hello students, you are watching Biology by Dr. N.S. Jain. Today we are going to study the part 2 of the nucleic acid, and today we will be going to discuss about the DNA. As you know very well that DNA was discovered by Friedrich Mischer from pus cells, and he gave the name nuclein. He gave the name nuclein. He has not used the word DNA. He has used the word nuclein. So DNA was first isolated by or discovered by Friedrich Mischer from pus cells, and he gave the name nuclein. And you must also know that DNA is polymer of nucleotides. Many nucleotides combine together to form DNA, and that's why DNA is called as polynucleotide. In the previous video also you have studied this thing, that DNA is a polynucleotide. And you also know that a nucleotide consists of nitrogen base plus sugar plus phosphoric acid, that is H3PO4, and the sugar in the case of the DNA is 5 carbon, and the nitrogen bases in the DNA are A, T, G and C. So a nucleotide consists of these three. Okay, now you must know that this is a sugar, okay, and this is carbon, 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 here also carbon, and here CH2OH, H, here is H, OH, H, H, OH, H. This is carbon number 1, 2, 3, 4 and 5. This is a pentose sugar, that is known as the deoxyribose sugar, which is consisting of the 5 carbon. You must know, before we will be discussing about the DNA in details, you must know about that where the phosphoric acid and where the nitrogen base is attached to the sugar. So always remember that the nitrogen base is attached to the C1 of the sugar. Always remember, nitrogen base is attached to C1 of the sugar, and it forms a glycosidic bond, and always remember that phosphate is attached with this one, right, with the 5th carbon. So phosphate is attached with the 5th carbon atom of the sugar, right? The phosphate is attached to the 5th carbon atom of the sugar, and it forms what? Phosphodiester bond. It forms what? Phosphodiester bond. Always remember, a phosphodiester bond is formed between two adjacent nucleotides, okay. Always a phosphodiester bond is formed between the two adjacent nucleotides. Just now we will be discussing more about the phosphodiester bond, okay. So this becomes what? This becomes a nucleotide, that is the nitrogen base plus sugar plus phosphate all combined together to form what? A nucleotide. Now let's have a discussion more on the DNA, okay. Now see here, when we are talking about the DNA, you must know about the properties of the DNA and other things also, okay. There were the scientists known as the Franklin and Wilkins. They used X-ray diffraction technique, and said that DNA is a double helix, okay, based on the photographs, based on the photographs from X-ray diffraction technique, based on the photographs from X-ray diffraction technique, Watson and Crick produced the famous double helix model of DNA in 1953, and for this the Nobel Prize was given in the year 1963 to Watson, Crick and Wilkins together. All these three were awarded Nobel Prize for giving the famous double helix model of the DNA. Okay, now you must know about some properties of DNA. Properties of DNA. Now, very first, the DNA discovered by Watson and Crick was bDNA. One thing you must remember, that DNA is of different types. Okay, DNA may be of different types. Say for aDNA, okay, bDNA, cDNA, ddNA and zDNA. These are the different types of the DNA. But the DNA which was discovered by the Watson and the Crick was the bDNA. You must remember this thing, that it is most stable form of DNA. So whatever the data and whatever the discussion we will be doing in this video will be strictly based on the bDNA. Okay, whatever the data we will be producing here, that will be particularly for the bDNA only. So the DNA discovered by Watson and Crick was the bDNA, and it is the most stable form of the DNA. Always remember that this bDNA is a right-handed DNA. It's right-handed DNA, that is, coiling of DNA is clockwise. Clockwise means it is in the direction of the clock, like this type. This is called as what? Right-handed DNA. But one thing must be remembered, that zDNA is left-handed DNA. zDNA is left-handed DNA discovered by Rich. It was discovered by Rich, okay, and its coiling is in anti-clockwise direction. Okay, so zDNA is left-handed DNA, but the bDNA what we are going to discuss here is a right-handed DNA, in which the coiling of the DNA is clockwise. Now, you must also know that the DNA is what? Double-stranded. DNA is what? Double-stranded. Now see here, DNA is double-stranded. DNA is double-stranded. Means it is also called as dsDNA. dsDNA means what? Double-stranded DNA. What does it means that double-stranded DNA? That DNA is made of two polynucleotide chains. The two chains form a double-stranded DNA, right? Like this type. A double-stranded DNA is of like this type. Like this type. Okay, this is the 3' end, this is the 5' end. Okay, Now see here, like this type. This is the 3' end. Okay, so this is a double helical DNA. This is what? A double helical DNA, and these are the two polynucleotide chains. These are the two polynucleotide chains. Okay, one thing to be remembered is that the two polynucleotide chains are running parallel to each other. They are parallel to each other, but they are not crossing each other. They are parallel to each other. You can see that the two chains are parallel to each other, but they are not crossing each other. Okay, they are not crossing each other. So DNA is made up of two polynucleotide chains. DNA is double stranded. Okay, exceptions are lying. Okay, exceptions are lying. Okay, the exception is pi x 174, right? It is having what? Single stranded DNA. It is having single stranded DNA. It is having single stranded DNA, and the single stranded DNA was discovered by a scientist known as Robert Schimmer. It was discovered by Robert Schimmer. Otherwise the DNA is what? Double stranded. Okay, now the two chains of DNA are antiparallel. This is very, very important property of the DNA. The two chains are antiparallel. What does it means? The two chains are antiparallel means one chain is running in such a direction, say for like this type, and another chain is running in this direction, 3' to 5'. Means both the chains are antiparallel. Antiparallel means what? Antiparallel means what? You can see that 5' end and 3' end are you can see that the two chains are not having like this, that they will be having 5' and 5' end. Okay, one chain will be having 3' end, the another chain will be having 5' end. One chain will be having 3' end, the another will be having what? 5' end. These two chains are antiparallel to each other. Always the two chains run antiparallel to each other. To prove this thing that the two chains run antiparallel to each other, we will be making a detailed structure of the two chains. Say for I am making sugar here. Say for this is a pentose sugar. Okay, and this is another pentose sugar. This is another pentose sugar. Now try to understand it. This is another pentose sugar. Okay, now these two chains are antiparallel to each another. This is what? Oxygen. This is oxygen. Now this is 1, 2, 3, 4, and this is 5th carbon. Okay, 1, 2, 3, 4, and 5. Now see here also 1, 2, 3, 4, and 5. So this is carbon number 1, 2, 3, 4, and 5. And you must know that the sugar is having at the third position of the sugar, a functional group is having the OH. A functional group is present known as the OH. Here also you can see that OH is present. Okay, so this is one chain. This is one chain. Okay, now what happens? That always remember a phosphodiester bond. A phosphodiester bond is present between the the two adjacent nucleotides. Means here you can see that at 5th position what will be forming? A phosphodiester bond will be formed. So 5th carbon atom of one sugar and 3rd carbon atom of the another sugar, you can see here that at the 3rd position, right, what is present? A functional group is present, and here a phosphodiester bond will be formed. So I am writing here that this is what? A phosphodiester bond. This is a phosphodiester bond. Okay, and you must know that at the 1st position what is present? Nitrogen base is present. Now the another chain is antiparallel to this. So like this type you can see that how it will be forming an antiparallel chain. So this is the another chain, right? Here also there is nitrogen base present. Now here it is oxygen. Now this is carbon number 1, 2, 3, 4, and 5. Okay, so this is 1, 2, 3, 4, and 5. Number this also carbon, 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 and carbon, and here also the carbon, 5th position. So 1, 2, 3, 4, and 5. Okay, So always remember that nitrogen base is attached to the first carbon atom of the sugar. Here also you can see that nitrogen base is attached to first carbon atom of the sugar, and these nitrogen base, in between the nitrogen bases is present what? The hydrogen bond. In between the two nitrogen bases is present what? The hydrogen bond, like this type. Okay, so always the nitrogen base is attached to the carbon 1 of the sugar. Now here also a phosphodiester bond will be formed. Now you can see here that a functional group is present here. A functional group is present here at the third position, that is known as OH group, and here the phosphate is present. Here what is present? Phosphate is present. Here phosphate is present. So again you can see that here a bond will be formed, phosphodiester bond. Phosphodiester bond. What does it proves? That this bond is present between two adjacent nucleotides. So this is a nucleotide, this is a nucleotide, this is another nucleotide. So in between these two adjacent nucleotide, phosphodiester bond is formed. In between these two nucleotides, what is formed? Phosphodiester bond is formed. So this chain is having direction like this type. Now see, this is the 3' end. So this is the 3' end, and here that is the 5' end. Okay, so here the chain direction, is 3' to 5'. Now see the another chain. Here third carbon is free. Here the third carbon is free. So here 3' end, and here you can see the 5' end. Okay, so this is what? 5' end. So we can say that these two chains are antiparallel to each other. So this proves that both the chains are antiparallel. So we have studied two properties. DNA is double stranded and it is made up of two polynucleotide chains, and the two chains of the DNA are antiparallel to each other, right? I can place nitrogen bases also here, है instead of the nitrogen base I can put here A, and I can put here, that is known as the T. Okay, I can put here G, and I can put here C. Okay, and these are what? Hydrogen bonds. So the two chains are held together. The two polynucleotide chains are held together by what? Hydrogen bonds. The two polynucleotide chains are held together by weak hydrogen bonds. These hydrogen bonds are very much weak. Okay, so these are weak hydrogen bonds. Now coming to the third property, that not only the two chains are antiparallel to each other, the two chains are complementary to each other. The next property is that the two chains are complementary to each other. Now the question arises, what do you mean by this complementary? Complementary to each other means a purine pairs with pyrimidine. A purine pairs with pyrimidine. Always remember, a purine pairs with a pyrimidine, right? A purine pairs with a pyrimidine. This is just to save the space, okay? Because pyrimidine is single ring structure, and purine is what? Double ring structure. Okay, so always remember that A combines with T, and G combines with C, means they are complementary to each other. Now what are A and G? A and G are purines, and what about T and C? They are what? Pyrimidines, right? So always a purine combines with a pyrimidine, and in between A and T, double hydrogen bond is present. In between A and T, double hydrogen bond present, and between G and C, triple hydrogen bond present. And that's why its melting point is high. Why? Because it has three hydrogen bonds, and that's why its melting point is high. So always the two chains are complementary to each other, A combines with T and G combines with C. A and G are purines, T and C are pyrimidines. In between A and T, double hydrogen bond present. In between G and C, triple hydrogen bond present, and its melting point is high because of the presence of the triple bond. Okay. Now, you must also know that if we talk about a DNA molecule, so in the case of the eukaryotes, you must remember this thing, that the DNA, that is the DNA is of AT type. DNA is of what type? AT type. Means AT content is more than GC content. A eukaryotic DNA is always having AT type of the DNA. Means in a eukaryotic DNA, AT content is more than GC content. And in the case of eukaryotes, linear DNA is present. While if we talk about the prokaryotes, then in prokaryotes, GC type of DNA present. In prokaryotes also, as well as mitochondria, as well as the chloroplast DNA. You must know that mitochondria is also having DNA, chloroplast is also having DNA, and prokaryotes are also having DNA. In them, GC type DNA present. Now question arises, what is this GC type DNA? Means GC content is more than AT content. Okay. And one thing more to be remembered is that in the case of the prokaryotes, the DNA is not linear, the DNA is circular. Okay. So you must know about a eukaryotic DNA also and a prokaryotic DNA also. Okay. Now let's have a look on the two chains of the DNA. Now see here, a DNA consists of two grooves. This is known as minor groove. This is called as minor groove. This is called as major groove. And again this is called as what? Minor groove. Okay. So you must remember that there are two grooves, minor and major. Minor grooves are shallow, they are not so much deep, while is very much deep. Okay. Now you must remember that length of one helical turn, right? You must know that length of one helical turn. You can also say that length of one pitch turn length. What is the pitch turn length? Or we can say that length of one helical turn. So the distance from one minor groove to another minor groove, forms the pitch turn. Forms what? The pitch turn, and the pitch turn is 34 angstrom. So either you can say length of one helical turn, or you can say that pitch turn length is 34 angstrom in the case of the bDNA. Now that is number of base pairs in one helical turn. Means in one pitch turn, how many nitrogen bases are present, how many nitrogen base pairs are present in one helical turn. So you must remember that is 10 BP, 10 base pairs. That is 1, 2, 3, 4, 5, 6, 7, 8, 9 and 10. So there are 10 base pairs. Number 3, distance between two corresponding base pairs. Now what are corresponding base pairs? Say for A, T, G and C. A and T is one base pair, G and C is one base pair. So what is the distance between this corresponding base pairs? Okay, the distance between corresponding base pairs is 3.4 angstrom. Why? Because length of one helical turn is 34 angstrom, and in one helical turn how many base pairs are present? 10 base pairs are present. It means distance between two corresponding base pairs will be what? 3.4 angstrom, that is divided by 2. Okay, now then comes to be diameter of DNA. What is diameter of DNA? Diameter of DNA means what? The distance between two phosphates. Distance between two phosphates. That is known as what? Diameter of DNA. So diameter of the DNA in the case of the bDNA is 20 angstrom. That is from here to here. That is known as what? 20 angstrom. I will be making like this type. I will be making the two polynucleotide chain like this type. A double bond T, G triple bond C. Here sugar is present, sugar. Here also sugar, here also sugar. And the bond present between one phosphate and two sugar is called as what? Phosphodiester bond. Okay. So this is the diameter. This is the diameter. So distance from one phosphate to another phosphate is what? The diameter of the DNA, known as what? The 20 angstrom. Okay. And the fifth property is distance between two sugars. What is the distance between two sugar? If the question is asked like this type, distance between two sugar means distance from this sugar to this sugar. Okay, so that is known as 11.1 angstrom. So I am writing here 11.1 angstrom. So these data, you must know about these data as also. That length of one helical turn, that is pitch turn, is 34 angstrom. Number of base pairs in one helical turn, 10 base pairs. Distance between two corresponding base pairs, 3.4 angstrom. Diameter of DNA, that is distance between two phosphate, is 20 angstrom, and distance between two sugar is 11.1 angstrom. Okay. Now, the DNA also follows an important rule, and that is known as Chargaff's rule, right? On DNA, a rule is applicable, and that is known as Chargaff's rule, right? Chargaff's rule. Now, what does Chargaff rule says? Chargaff rule says that percentage of A is equal to percentage of T, and percentage of G is equal to percentage of C. Means Chargaff rule says that A is equal to T and G is equal to C. A is equal to T and G is equal to C. This is the first point of the Chargaff rule. But A plus T is not equal to G plus C. Chargaff's rule says that A is equal to T, G is equal to C, but A plus T is not equal to C. Also, you must know that A plus G is equal to T plus C. A plus G is equal to T plus C must be always 50%. And this is very important to solving the questions. A plus G is equal to T plus C must be 50%. Sometime a question is asked, if G is equal to 30%, what is the percentage of T? So how to solve this question? We know very well A is equal to T and G is equal to C. So you must write here A plus G is equal to T plus C. Now G is given here 30%. So we know very well G is equal to C. So automatically the C will become 30%. And we also know that A plus G must be 50%. So to make A plus G 50%, we will putting here what? 20%. So 20 plus 30, that comes out to be what? 50%. And we also know A is equal to T. So if A is 20, then automatically T is what? 20%. So T plus C will also become what? 50%. So A plus G is equal to T plus C. So what will be the answer for this question? What will be the percentage of the T? So the percentage of the T will be 20%. Like this type of the questions are asked many times in medical entrance examinations. So you must know this thing. Also, you must remember this thing. Also, Chargaff rule says that A plus T upon G plus C is equal to base ratio. A plus T upon G plus C is base ratio. Remember that always you have to remember this thing. A plus T upon G plus C is equal to what? Base ratio. It is less than 1 in prokaryotes. It is less than 1 in prokaryotes. It is less than 1 in prokaryotes. And it is more than 1 in eukaryotes. It is less than 1 in prokaryotes, and it is more than 1, and it is more than 1 in the case of the eukaryotes. Now why this happens? So you must remember that in prokaryotes it is less than 1. Why? Because prokaryotic DNA, because we have just now studied that prokaryotic DNA is GC type. Now what will be the impact of the GC type of the DNA? GC type means what? The lower value will be more in the case of the prokaryotes and upper value will be low. Okay, so we have to impose what? 0 point something will be the value coming, which will be less than 1. While in the case of the eukaryotes, the eukaryotic DNA is AT type. So what will happen? What will be the impact? The upper value will be higher and lower value will be lower. So always when we'll be cutting the upper value, we'll be getting the value 1 point something, which will be always more than 1. So Chargaff rule says A plus T plus G plus C is equal to what? Base ratio. And also you must remember that base ratio is constant in a species but varies in different species. It is constant in a particular species but it varies in different species, means different species are having different base ratios. Okay, now one question is asked many times: what will happen if DNA is boiled at 80 degrees Celsius temperature? What will happen? So what will be the answer? That denaturation of DNA will occur. Denaturation of DNA occur. What is denaturation? Means double-stranded DNA is changed to single-stranded DNA. Why? Because on boiling, the hydrogen bond will be broken down and the double-stranded DNA will be converted into single-stranded DNA. So what will happen if DNA is boiled at 80 degrees Celsius temperature? So you will say that double-stranded DNA will be changed into single-stranded DNA. And if what will happen if DNA is cooled again? So what will be the answer? DNA will come back in its original state. That is, it will become double-stranded. It will become what? Double-stranded. And this process will be called as what? Renaturation. This process will be called as renaturation. So if you boil the DNA, then double-stranded is converted to single-stranded DNA. This is called as denaturation. And if you cool the DNA again, then the DNA will come back in its original state. From single-stranded, again it will become double-stranded, and this process is called as what? The renaturation. This process is called as what? The renaturation. And at the last, you must know that DNA is the only living molecule. You must know that DNA is the only living molecule which has power of replication. Replication means what? Making copies of its own type. So dear students, this was all about the DNA. This was the part 2 of the nucleic acid series. Keep watching. We'll be discussing more about the nucleic acid. We'll be discussing more about DNA. We'll be discussing more about the RNA in the upcoming videos. Keep watching my videos. Thanks a lot.